It has a 5 cap which allows it to maintain stability. snRNPs contain snRNA surrounded by a protein coat. snRNPs, along with other protein, form a spliceosome. Inside the spliceosome, snRNA binds by the base pairing of different nucleotides at the intron-extron junction. snRNPs function as ribozymes, which cleave off the breakage of an intron. This intron becomes degraded, along with other spliceosome components. What's left is an mRNA molecule. The mRNA molecule contains a poly-A tail that's attached to it. The poly-A tail allows it to maintain stability and lead the nucleus to the nuclear pores. Activator proteins bind to distal control elements, which are grouped as an enhancer in the DNA. A DNA bending protein bends the DNA so that the binded activators can come closer together to the promoter. Nearby are general transcription factors, mediator proteins, and RNA polymerase II. The binded activators bind to different mediator proteins and general transcription factors to create a transcription initiation complex. Regulated synthesis of repressible enzymes in E. coli. When the TRP operon is usually on, which means that it has an inactive repressor. When the repressor is inactive, RNA polymerase cannot synthesize the operon's genes and cannot attach to the promoter. When RNA polymerase can attach to the promoter, RNA synthesis occurs, and an mRNA is created from the TRP operon. When an mRNA is created, it forms 5 polypeptide subunits. The 5 polypeptide subunits then create enzyme, which eventually forms the TRP enzyme. Shows the regulatory synthesis of enzymes of the lac operon in E. coli. When lactose isn't present, the protein is active. When the protein is active, it binds to an operon. When the protein is bound to an operon, RNA polymerase cannot travel along a strand of DNA and synthesize and start RNA synthesis. When lactose is present, a protein, along with allolactose changes the shape of the protein, causing it to become inactive. When the protein is inactive, it cannot bind to the operator because it's changed shape. So RNA can travel along the DNA strand and form enzymes. The LAC-Z operon is responsible for galactoside. The LAC-Y enzyme is responsible for permease, and the LAC-A operon is responsible for transcetyl. The LAC-I operon is also responsible for the protein.